Hey there, science enthusiasts. It's Bill Nye here, ready to take you on a wild ride through the microscopic world of DNA. DNA is like the instruction manual for your entire body. Each recipe in this cookbook is called a gene. These genes tell your cells how to make proteins, the building blocks of your body. Imagine shrinking down to the size of a molecule and zooming into one of your cells. You'd see DNA coiled up tight like tangled shoelaces, unwinding when it's time to use the information. Your cells constantly read this DNA cookbook to make proteins, helping your body grow, repair itself, and stay alive. Pretty cool, huh? But how exactly do cells read DNA and make proteins? All right, science friends, let's talk about translation. DNA translation is like decoding a secret message, changing DNA language into protein language. Think of DNA as written in one language, DNAese, but your cells need the instructions in proteinese. Translation is the process of changing the DNA language into the protein language. Proteins are the workhorses of your body, doing all sorts of important jobs. Some proteins give your body structure, like keratin in your hair and nails, while others, like enzymes, help chemical reactions happen. Without translation, your cells wouldn't be able to make these crucial proteins. It's happening in your body right now, millions of times every second. Isn't that amazing? Now that we understand why translation is so important, let's dive into how it actually happens. The first step in this incredible process is called initiation. Initiation starts when a ribosome attaches to the messenger RNA, mRNA. mRNA is like a copy of the DNA recipe sent out of the nucleus to the cell's protein-making factory. The ribosome reads the mRNA and helps build the protein. It looks for a special start signal, like the "begin here" instruction at the start of a recipe. Once the ribosome finds this signal, it's ready to start making the protein. Transfer RNA, tRNA, brings in amino acids, the building blocks of proteins, starting with methionine. All right, science pals, we're now in the thick of protein production. Elongation is where the real action happens. The ribosome moves along the mRNA like a train on a track, reading a set of 3 letters called a codon. Each codon corresponds to a specific amino acid. As the ribosome reads each codon, a matching tRNA brings in the correct amino acid. The ribosome then helps attach this amino acid to the growing protein chain. This process repeats with the protein chain getting longer and longer. The ribosome keeps chugging along the mRNA until it reaches a special stop signal. This signal tells the ribosome that the protein is complete. We've made it to the final step of translation termination. When the ribosome reaches a stop codon on the mRNA, it's like hitting the brakes on a train. This special codon signals that the protein is finished. Special proteins called release factors come in to help. These release factors help separate the newly made protein from the ribosome and the mRNA. The finished protein then goes off to do its job in the cell. It might become part of a muscle fiber, help digest your food, or carry oxygen in your blood. And just like that, your cells have successfully translated the DNA recipe into a working protein.